All right. Well, I'm going to make sure that folks have the chance to join us. All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide. This webinar is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support make this webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code %10 to receive 10% discount on your registration. ASPB members are able to take advantage of so many other benefits in addition to being able to support this webinar. So you can learn more about ASPB and all the opportunities we provide at aspb.org. Next slide, please. This webinar is recorded, and so we've had so many other wonderful and informative webinars that we've done over the years. So if you're looking for more content, be sure to check out the Plantae YouTube channel, where you can watch all the exciting talks that we've done. And so make sure you find some time to tune in. And also, if you see something interesting, be sure to kind of share the wealth, and so pass it along to your colleagues and friends. Next slide, please. Registration for Plant Biology Conference, which is hosted by the American Society of Plant Biology, is open, and so it's taking place in Milwaukee this year from July 26th to the 30th. It is a very awesome networking event. It's uniquely positioned to bring together members of the plant science community, where you can acquire knowledge, network, learn new skills that will be necessary for your work and helping us advance plant science. And it's also really beneficial for all career levels, so all are welcome. And so if you want to learn more and want to get registered, you can learn more and register at plantbiology.aspb.org. Right now, we are still running the advanced rate. And so if you register by June 10th, you're able to get some pretty good discount on your registration. So make sure you tune in and also get registered as soon as you're able. Next slide, please. Before we get started, I'd like to share a few housekeeping items to make sure that you get the most out of your attendance in today's webinar. If you're experiencing technical issues, let us know about those in the Q&A box, or you can email me directly at jpadilla@aspb.org. If you have questions for our guest speakers, you can add them on the Q&A box. During our Q&A, after all the talks, the moderator will be kind of moderating all those questions and getting answers for those questions, at least as many as we can from our speakers. If you have trouble connecting or need to leave the webinar early, as I mentioned, we are recording this webinar and you'll be able to watch it once it's processed on our YouTube channel. So if you're not already following the Plantae YouTube channel, make sure you do so. And so without further ado, now I'm going to turn it over to our Plantae Fellows, who will be facilitating today's discussion. So with that said, take it away, Plantae Fellows.
Hello, everyone. Welcome to today's webinar on AI in plant biological science research, which is a very interesting and relevant topic in today's time, as we all know. So the objective behind organizing this webinar was to bring in people from various backgrounds, which you will see as we introduce our speakers, so as to understand how AI is playing shaping plant biological research in current times and how it's going to help us in the near future. So the outline for the webinar goes as such, as I have just made a general introduction to the webinar, will be followed by the speakers' introductions shortly. And then once the speaker finishes, we'll have a Q&A session. You can put in your questions in the chat box, in the Q&A box, and then we'll conclude our webinar. Next slide, please. So, as I mentioned earlier, we have brought in speakers. Our distinguished speakers are both from academia and industry. And we also have someone who is a senior researcher in the academic field who is just like us, one of us who can, who can really elaborate us as to how beginners can work with AI in research. So we are very pleased to have Dr. Pau Formosa Jordan, who is a principal investigator with Max Planck Institute for Plant Breeding Research in Germany. We are also pleased that Dr. Marina Kuzmenko has agreed to be here with us as a speaker. She's the CEO and co-founder of PTO, which is a UK-based agri-tech company specializing in AI research, and Dr. Manish Tiwari, who is a senior postdoctoral scientist at University of Wisconsin-Madison, US. Next slide, please. With that, just a small introduction. I am Indrani. I'm a current ASPB Plantae editor, and I just concluded my ORISE postdoctoral fellowship with USDA ARS. I was working with chocolate plant disease resistance against fungal pathogens, which are well known to be creating a lot of devastating diseases in chocolate plant. You can find me my socials are given here, and yeah, I'll be the moderator for this webinar, one of your moderators for this webinar. Next, please. With that, I would like to introduce our first speaker, Dr. Marina Kuzmenko. She is the co-founder of PTO, a UK-based agri-tech company which focuses on implementing AI applications in agriculture. She holds a PhD in business law from University of Kyiv, Ukraine. She has successfully led projects which have been funded by Innovate UK and has represented her work at various international events. She's also a regular speaker at various educational events like this one. And through her, we are going to know how AI is currently being practically used in industry, through industry in research. With that, I would like Dr. Marina to start her session, please. Thank you.
Thank you so much, Indrani, for this nice opportunity. I would like to share my screen firstly, and I will make it in full screen mode, and I hope you can see it now. So can you see my screen? Is everything all right? I hope so.
Yep, we can.
So
yes, we can.
Thank you. Thank you so much for confirmation. So let's go briefly, let's speak briefly about this talk which I will have today. First of all, I would like to give you the context. I know that you know about the context and how the situation is about practical application of AI in plant science, but at the same time, I would like to focus you on some specific points which maybe you have in your practical work and could be these issues could be solved with what I will have next. And in next part, I will show you the four specific directions where the AI currently is applying. And finally, we will also speak about challenges because obviously there is a lot of opportunities, but the reality is sometimes different. So let's go to our first slide. And it's, by the way, in the corner of this slide, you can see the reference. So everything which I have demonstrated here, you can just double check and go in depth, because obviously 15 minutes, it's quite short period of time to dive into every of these researches because they are absolutely interesting and they are relevant for today's topic. So I encourage you to read them because I found them truly interesting, and these illustrations which I picked up, I believe they will help you to make this logic chain of where we are now and where we can go in the future with AI. So example of plant, and I'm sure we all know that plants are alive and we work with them every day, and we know that they are reacting on everything which they have experiencing. But the problem is that they are silent. We are not speaking about bioacoustics now and other areas which are truly interesting, but they are not practically so widespread as we wish. So what can help us to hear the voice of the plants? And this is exactly where we believe that artificial intelligence will be the most helpful, because artificial intelligence helps us to make the existing data, which is different data, which is visual data, numerical data, audio, or any other type of data. Sometimes it's temperature measurement, sometimes it's satellite imagery or anything like that. And we can understand it, and we can implement it into understanding how do the plant reacts on something. We can get the feedback from plant when we need it to understand, are we going the right way or not? And I would like to show this photo. And this is Dr. Hong Luo from one of the universities in the United States of America. And on the place of him, every of us could be, because when we are doing any type of experiment, first of all, as usual, we are using our eyes and its natural ability. But the problem is that we are limited. And the limits are not just about our eyes and our brain, which has memory capacity, and we have some span of our memory, but also it's about the time which we have for doing specific experiment. It's about the personal attitude and personal judgment which we are putting into the summary which we are doing. For example, you can see two plants and can say that, okay, those on the left hand side is a little bit more greener than the on the right hand side. And I will stay next to you and I will think, oh no, I think they are the same. So how we can quantify this personal judgment, personal opinions, and remove this subjectivity, but bring more independent, some independent arbiter into this situation. And also it's about the scale, because if we go back, so you can see two plants and you can compare them. Even 100 plants, it's still fine. You can have your colleagues involved in this work, and you can do this manually. But if you have, let's say, thousands of plants, and you need to do the measurements to make your to enrich your research with data, you need to do measurements on daily basis. But actually, you also have some other stuff to do, and you need to write everything and record everything. So it's a lot of problems with scaling the experiments because of the physical borders. And that's why artificial intelligence is the empowerment which is trying to remove these borders. And it helps us to make these experiments bigger, wider, in more varieties, in more traits. It's just add word more. So that's why how we can process now, if we are not speaking about this digital technology, AI technology, how we can process plant phenotyping, plant genomics, in field data, and remote sensing in one go. Yes, we have human abilities. We have 4 people who are responsible for that. And yes, on this stage, I don't say that we need to remove these people, no. But if we give these people the chance to be more productive and to use their knowledge in quicker way, using the speeding up process based on AI, this makes everything faster. And that's why we can do this speed breeding experiments. We can do this, all the experiments which were limited in the past, but now they are the door is open. And I believe that introduction of generative AI, which has happened a few years ago, starting from just textual generating text like ChatGPT, Gemini, Claude, and some other models which are publicly available. And then when we are going to the image generation, audio generation, video generation, so you see the speed of AI is very high, but as of now, it's more about the let's say, generic context. When we are speaking about practical application for niche, because plant science is a very niche direction, then I would like to point your attention to 4 specific directions, yes, for specific ways how AI is practically applying and what can be helpful. So let's start with first one, and this is actually what we are practically doing, because this is a screenshot of one of our applications. We are in this application, we are helping one of the scientists who was breeding the soybean variety. He had a task to calculate, to count the root nodules. And this is such a manual and time-consuming task, you know, when you have this very nice root and you need to sit down and just count and write, and the next image and you are just again count and write. And then he asked us, is it possible to automate this? So based on his 10 photos, we just did this automation. And yes, we didn't charge for this because we honestly wanted to help him and just try. Is it possible at all? Because we have read in previous work that in previous time that there is already existing research paper that helps to understand how to count nodules, but we were thinking, is it possible just in practical level if we are not involved in this huge lab and everything? And this is like a proof that yes, it's possible. So we have counted them. Obviously, this model which we have developed, it's trained specifically on these images which we got from the scientists, but the beautifulness, the beauty of AI is that you can retrain model. So based on this model, if we get another photos, let's say they will be not on this blue color background, but on yellow color background, I don't know, red color background, it's still possible. If the nodules will be bigger or smaller, it's possible just to train the model based on new imagery input, and the model will already know on blue background and another background as well, so it will be even smarter. And the same way is working with any type of plant phenotyping. When you are doing, you need to count flowers, or you need to, let's say, measure flowers and compare them or something like that, which is, you are just now using eyes, and it's possible to do computer vision, which is like computer eyes to do the same job and provide information in quantifying and qualitative way. The next thing, which is also interesting, is about just let's imagine that I think many of you, maybe probably every one of you, have tried generative AI. So, and you see how it's quickly providing the output and the providing answers to your questions. But let's imagine there is like kind of generative AI for omics databases, and you just put your prompt and you got the answer regarding the like how they will work together, or how they will what the result will be after we will have this specific architecture. And this is the point that all this gen production, it's currently yes, we can think and predict and do some modeling, but at the same time, empowered with AI, we can get more ideas, let's say, as well as another nice thing. Even now, when we have, as I already mentioned, different models available, it's already possible to compare the results. And this is one of the messages I wanted to put it at the end of the presentation, but I want to say now, please, if you have a chance to compare the results which you got from AI model, compare it, and don't trust just one input which you on output which you got from your input from your prompt. Another interesting direction, so we have already named two, it's the third one is about the analysis for stress, or analysis for disease response from the plant. This is again the real example. So we have, it was not the scientific example, but I am sure that it will be relevant for you as well. It's a farming, one of the farmers, he had the cucumber crops. So he had the control line, control plants, and then he has 5 other lines with 5 different treatments, and he wanted to understand the early chlorosis, presence of early chlorosis on his plants. But the problem was that he has many of plants, and he was standing in front of his plants, and he was saying that, yes, I understand that these are kind more yellow, and I saw also that this is working better than those one, but just how to explain this in numbers. So what we have done, the farmer recorded the video, ordinary smartphone, recording video 1 minute every row. And then we took these videos, we labeled all these plants, not all on this video, but we trained the model to understand where is the plant and where is not the plant. And we actually put all these videos through the model, and model has discovered 4,228 plants, leaves, which he analyzed. Not he, but model analyzed. And then basically it was the comparison also about the greenness, because there was a threshold when we got understanding that this kind, like the where we need to understand the plant is green, and everything is which is below is considered as yellow and the presence of chlorosis, and what is above, it's okay, it's healthy. Based on processing of these 4,000 leaves, we were able to compare and provide him the information about his treatments, and he was like, he didn't just guess, he had some proof of his knowledge. And final direction, which is relevant regarding application of AI in plant science, is about the modeling, but it's about the like purely theoretical part. So in this case, we are just trying to forecast future, but as I mentioned, forecasting modeling already exists a lot. The only thing that we are speaking about more productive forecasting. So instead of just one model, which like all the team of 10 people is building, now 10 people can build 20 models, and they can compare it, and from this 20 model, they can find like 3 with the best case scenario, the good basic case scenario, and the worst case scenario. So this is about, as I said, the word more productive. At the same time, real life, real experience. I think many of you have been in rural areas, and even sometimes it's not about even rural areas, but about the connectivity. So what we have experienced in as a first question when we are trying to do practical application on, let's say, breeding experiment far from the town or the city. And the question of internet, is it available or not? Because, for example, when we are speaking about especially real-time processing, it's even a separate question. I will not touch it because, yes, there are opportunities to do it in real time, but it requires huge investment into the equipment. But if we are speaking even about the processing, like sending to cloud and getting some response, so it still requires stable connection. And what we see from existing situation, that it's not so good situation, and the situation is challenging specifically in global south, if we are speaking about the global scope. And for example, in this case, I would like just to tell you that we have developed this mobile application for leaf area measurement. It's called PTL Pro. We have other parts of this mobile application, but the interesting thing about this application that it's small, but it can work in rural area and without internet. And this is the nice thing that yes, AI sometimes, if we are speaking about very memory consuming and energy consuming tasks, it's quite challenging to deploy them in field or do it in real time. But if we speak about some niche but small tasks which still are needed, you still need this data, and especially if you're in the field, it's so nice to get it right here, right now. So there are already existing tools, AI tools, mobile tools, which can help you with, as I mentioned, these very narrow tasks. Because if you will go outside and just ask someone how to measure leaf area, they will just look and they didn't understand. It's only plant scientists who know about this secret. Another thing is about data. And again, about practice. So we are working with one interesting team from Ghana, regarding the cocoa beans, and they are trying to collect the data. And the problem is that it's always a challenge. It's a challenge, as I mentioned, data transition. It's always a problem because, unfortunately, what we discovered that in Africa there is not so strong connectivity for these purposes. But also regarding the data collection, it's always about protocol, about the light. How much light do you have for taking the image of this specific plant? Where do you need to stay? Then what type of camera are you using? And how good is the quality of the imagery which you are generating? Is it from smartphone, or is it from any action camera, or is it from any type of camera? So it's very important to get the good data, because data is a fuel of any AI model. So if you, let's imagine you have a very good model and it's very nicely trained on everything and like almost ideal, but you have poor input. I mean, you have the photo which has taken with blurred quality, or let's say the image is covered with shade, and you are asking something very specific regarding, let's say, area of this plant, leaf area index of this plant, then you may get a response, but the problem is that this response will not be reliable. And this is the challenge, that you need to invest into your efforts, your time, into collecting good data. The second thing is here, and demonstrating you that actually your knowledge in plant science are unique. Because when we speak about training the model, AI model, it's always about that there is some guy who know where is a leaf and where is not a leaf. I'm just generalizing, but if we are speaking about some genomics, your knowledge is just decisive, and you are just deciding, is it true or not true? And based on what you are labeling means this, putting these small boxes, bounding boxes around the plant, or around the specific feature, and saying the computer that this is something. So this is called labeling. So that's why labeling is also very important, and qualified knowledge of the person who is labeling the data set, which is then is fed to AI model. It's also very important. And also, yes, this is I'm slowly moving to the end of my presentation, but this is the core message which I want to say you, that it sounds like a very technical thing. But again, thanks to Indrani, she said that I am not an engineer at all. I am the person who worked with engineers. And that's why we, speaking about the future of AI in plant science and plant biology, we need to understand this is the collaboration, this is the friendship of engineers and biologists and scientists who are knowing about the plants. And also one more important thing I mentioned at the beginning. I am not I'm not saying that we will be swapped with AI. However, there is one of the theories that say about that. But my message is that we can be empowered by AI, and this is what we are providing, and what we are believing in. So please don't be afraid. Learn as much as you can, and yes, I am always happy to speak with you, because I'm present on LinkedIn and X Twitter, which is called X currently, and very glad to continue our discussion and also answer your questions after this this talk. Thank you.
Thank you, Marina. Thank you for such nice talk. Now we will go to our second speaker. First, I introduce myself. I am Gaurav. I am a Plantae Fellow for this year, and I am doing PhD at Max Planck Institute for Plant Breeding Research, the same institute where Pao is a group leader. Next slide, please. Dr. Pao Formosa Jordan is a group leader at Max Planck Institute, and he completed his PhD in physics, and then he moved to biology at the University of Cambridge, And now he works in integration of the aspect of modeling and biology and answers fundamental questions like how cell actually differentiate and how should typical meristem take decision whether it should make leaves or flowers. So now I welcome Pao so he can share his screen.
Thanks, Gaurav, for the introduction. Hi, everybody. Do you see my screen correctly?
Yes, we do. Yes, we do.
Okay, good. Okay. Good morning, good afternoon, good evening to everybody. I'm really excited to be here today. Thanks to the Plantae organizers for inviting me. So today I'm going to talk to you about how we use AI to understand plant development. Mainly, we use it to understand images at the moment. In my group, we have different backgrounds. We are mathematicians, physicists, biologists, engineers. And the approach that we have is what is called plant computational morphodynamics. So we use microscopy. So we try to have images of seeing plant cell walls, the cells, the nuclei, and you name it, the fluorescent reporters of your interest, of the processes that you want to understand in plant development. And we perform quite a lot of image processing and image analysis. So we might spend half of our time in working on the images. And then we also try to build a theory and also build models to understand plant development, right? So there's this feedback between theory and experiments. And where is the AI here? So for now, it's in the image processing and in the image analysis part, but we hope to use it more in microscopy itself and in modeling. So actually, AI has been very, very important in the previous years, as you might know, in the field of image processing. So the classical methods that people know, such as edge detection and watershed algorithms to segment on one side blobs and on the other side, for instance, cell walls or cell membranes are still very useful, but there's new technology from AI that are making things much simpler, such that we can deal with images in a more high throughput way. I should cite, for instance, that in 2019, there's been the software called Elastic that was made by people in Heidelberg, so the group of Fred Hambrecht and Anneke Schut. And this software is very useful and has very good pipelines that are easy to learn. For instance, a very popular one is the use the pixel classification, that it's very easy to train. So you do some brushes in your objects of interest and also in the background, and then you can train using a random forest very quickly your classifier, and then you can segment your objects of interest very, very quickly. And that's a software that we use a lot in our lab. And even more recently, there's been, I must cite the paper of Wolny et al. From the group of Anneke Schut in Heidelberg. So this is about the software that uses a neural network to do predictions of the boundary of your cells. And then afterwards, you can apply segmentation to do your segmentation in 3D, right, of your system of interest. So now there's these and other softwares that are being more and more useful. And I'm really very excited to see that this is making our life easier and easier. So on the research question side, a big part of my group tries to understand how do you form cellular patterns. So here you can see a bunch of cells that would be presumably undifferentiated. And then after growing and dividing, you get patterns of different cell types. So how do you achieve these cellular patterns? And obviously, AI can help us to characterize this process and define the different cell types during development in the different tissues of interest. So a tissue that we've been looking for several years now in collaboration with a Rother lab, it's the sepal of Arabidopsis, in which you can see these very big cells, and these are called actually giant cells. These giant cells have a huge nucleus that has many copies of the same chromosomes, and that's called the fact of being polyploid. So you have several copies of the chromosomes. And it forms a very interesting pattern. And the question that we had is how just some scattered cells become giant while others not. Okay, so I'm not going to have time to talk about this, but you can check in our papers. But perhaps I can give you some ideas of how we characterize the images that we get. So here, that would be an image from our collaborators, the Rudder Lab, and then we can segment it. We're using morphographics, which is another software that you can do watershed segmentation, classical method, but also you can start doing AI here, segmentation. And then you can perform supervised cell type classification, meaning that if you take a part of your image, you can annotate which cells are which. Okay, you do it in a few cells, and then you take your algorithm, your classifier of interest, to perform the classification. Okay, so I have to say that this can be easily done in MATLAB. You have different algorithms that you can choose, and it's very easy to use, and also in morphographics. And in our case, that's a simple classification case. You can use even what it's called support vector machine, the quadratic one. It allows you to do a very good classification with a very good accuracy of these giant cells and these two other cell types that you can see, which are called pavement cells and stomata. Good. So we have patterns of different cell types in many, many tissues. And actually, recently, we team up and we are part of NSF BII Integration Institute, which is called the Polyploid Integration and Innovation Institute, with many groups in the US, that we try to understand how polyploidy affects tissues, organisms, and so on and so forth. And so the fact is that many plants and actually non-plant tissues exhibit spatial patterns with cells having different ploidy. So it means that different cells have different amount of copy numbers of chromosomes, right? And for instance, in the sepal, the giant cells can have 8 copies of the same chromosome, or 16 or 32 even. And also this would happen in the leaf. That's a portion of the leaf. So some cells have more copies of chromosomes, some others have less. So that's we see that really ploidy is patterned. And this is also well known that in the root, you have ploidy that is patterned also throughout different layers, right? And there's other examples that I can put you. But the fact is that we thought, okay, then that's it's relevant in development and also in disease. So we should start quantifying this in a high throughput manner. There's methods, but can we use AI to make things more high throughput? So ploidy, the number of copies of chromosomes, should be related in a way with nuclear science. That's something that has been studied. So then we can use images in which we see with our dye or our reporter of interest that relates with DNA content in one way or another. We can use these images and start doing this in a high throughput way. So that's what we did together with the Rudder Lab and the Fox Lab. And in a nutshell, here you would have your 3D confocal image. In this case, we are looking at a reporter that is related to histone, that is related to DNA content, right? And then we use the pixel classification with Elastic. We can do 3D nuclear segmentation, and then we use unsupervised Gaussian mixture models to have our ploidy map. And I'll explain this in a second. And this pipeline is called inferring spatial ploidy, iSpy. So in a nutshell, if you have your single cell data quantified, you name your feature of interest, could be nuclear volume, nuclear projector area, fluorescence intensity, you will get this kind of histogram in which you see different peaks. These peaks are presumably the different ploidies of your different cells. And then you can try to find the different groups of cells that you have. You can try to fit different Gaussians, right? And how many Gaussians you can fit there. So the cool thing is that you have methods nowadays that decide for you. I mean, nowadays, and it's actually an old method. You can use information criteria such as the BIC or AIC that will tell you what's the optimal number of Gaussians that you could fit there. So that's why it's an unsupervised learning method. So for instance, if we look just at the nuclear volume, we would have this kind of histogram, and the two first peaks would correspond to the populations of cells that have 2 copies of each chromosome. The second peak would be 4 copies of each chromosome, so on and so forth. But also you can do the same exercise looking at the fluorescence intensity of your reporter, and we have somehow equivalent results with some differences. In both methods, we have that the optimal number of Gaussians is 6, and you can try to combine nuclear volume and nuclear fluorescence and then do what is called the 2D Gaussian mixture model fit, okay? And that would be the result of your groupings in these different colors. And then you can try to compare what's going on when you have fit using just the volume, fit using just the intensity, and the fit using the 2 characteristics, so that will be the 2D fit. And then you can compare it with your ground truth, and in our case is the flow cytometry data in green, okay? And if you look carefully through this plot, it will be easy to convince you that the 2D fit is doing a good job. It's looking close to the ground truth and has less error bars. So from this, we can finally plot our spatial ploidy map, okay? So we try in these Arabidopsis cotyledons, but also we can try to apply it with to other tissues. So the Fox lab is expert in Drosophila, and if you injure the Drosophila digestive system, you will induce polyploidy there, and then you can run the pipeline, and then you will see that in the mild injury and severe injury, you are having higher ploidy cells, and there's a gradient of this pattern between the anterior and posterior axis. And actually, we also tried our pipeline with the Fox lab in heart data from humans. So the human heart, when it ages, it creates also polyploid cells, and that would be the images of left ventricle and left atrium. So our pipeline is new. We are just having this in the bioarchive, and we hope that people can try it. So in summary, what I told you now is that image analysis is benefiting a lot from AI. We should all keep an eye on its progress and use the new tools to save time and find very good and relevant discoveries. Supervised and unsupervised learning methods, some of them are old, but it's good to try to use them to easily understand how tissues are patterned and developed. And I would like to advertise again our new tool, which is called iSpy. You can find it in bioarchive and also in GitLab. You can scan this QR code, and please try it in your own systems, and we're happy to help you and to receive feedback. And then finally, I would like to thank the work to my wonderful group, also to my collaborators in Cornell University, in the Rother Lab, in the Fox Lab, also the Polyploidy Institute, and also the funding sources, and the Max Planck Institute, of course, and our department, and especially thanks to Nicholas Russell, which is the main driving force of the iSpy. Thanks for your attention. I should stop sharing the screen, or
all right. Thank you so much, Pau, for excellent presentation. Let's move to our next speaker. Before that, I am one of the moderator, and I will introduce myself. I am Ved Prakash, and a postdoctoral researcher at Ohio State University here, and working with plant-virus interaction. So our next speaker is Manish, and he's a postdoc at Department of Bacteriology at University of Wisconsin-Madison. And his research centers on symbiotic interactions between plants and organisms, with a particular focus on root nodules and abscular mycorrhizal associations. He's especially interested in how these symbioses respond to biotic stresses given by climate change. And he loves playing pickleball, soccer, cricket, and loves to be active. Manish, stage is yours. I will stop my screen and you can share.
Thank you, Ved. I guess everybody can see my screen?
Yes.
Thank you, Marina and Pao, for such a wonderful presentation. I really learned a lot from your presentation, and I am going to present a perspective of a graduate student or a postdoc or an early career researcher. I'm not going to make a model. I am going to see how we as early career researchers can use the models you guys are making. So we are kind of the users of the models, these models which has been made by Pao or Marina. So I'm going to talk how we can harness the AI in biology, particularly in our research, and a perspective from the graduate student or a postdoc or early career faculty. So already Pao and Marina has given a lot of introspection how we can use the AI in the biology, but I'll just to summarize, we can use AI for genomics and proteomics. For example, discovery of a gene network and a protein network, how they can influence a particular biological questions like stress or nutrition. We can use the AI to predict the structure of protein. Everybody might be aware of AlphaFold DB, which is a deep mind based program to predict the structure of the protein. And if we talk about the plants, you can use AI to predict a bioactive compound or a ligand for a receptor. And as Marina showed in her presentation, that you can put the images and train a model to predict if your plants are diseased or healthy. And in agriculture, we use a lot of AI-powered tool for phenotyping, as shown by Marina, or as Pao showed, you can check the ploidy levels in the cell, or you can use it for a better breeding. I'm going to talk about some of the AI tools that I regularly use, like almost you can say every day in my research. So I'll start with the ChatGPT, which is I assume everybody knows about that. And apart from like asking even very general questions, apart from my research, I use ChatGPT for coding help. I take help from ChatGPT for coding. And then I have several evidence-based literature review platforms like Elicit, Consensus, Notebook, Perplexity. And since I'm not a native speaker, sometimes I need programs so that I can improve my scientific writing. And for that purpose, I use Grammarly or DeepL. And then there is a program called Research Rabbit where you can get a complete view of the like mapping of the articles related to the scientific questions you have asked. For making presentations, I take help of Canva or Gamma app. And as I told, AlphaFold DB is, I use it for protein structure prediction. And BioRender has recently input AI in their app so that you can use this AI edit feature to design visuals or make plots. And I use ImageJ to analyze the microscopic images, as you might have seen a similar program, iSpy, developed by Pao. So these are some of the tools, AI tools that I regularly use in my research. I'll start with how I use ChatGPT in my research work. So first of all, if I have to ask just a broad question, like give me an overview of the topic, it could be related to research or does not relate to research, but I can ask any broad question to ChatGPT and voila, you can get an answer. Or if I want a summary of a research article, I can just input the detail of the research article and I can ask ChatGPT, please summarize me this research article in a simpler form. Sometimes you are not a native speaker and you might be feeling difficult to understand the jargons of the scientific literature. So in that case, you can use ChatGPT to summarize a research article in a simpler form. In my research, sometimes I need to refine my hypothesis, or if I have some data, I want to ask what could be the relevant research questions which I can pose. So in that case, I can use ChatGPT. I can generate the outlines for paper or grant when I write a grant proposal, and if I have to show the broader impact, or if I have to get the outline of the grant, I can ask ChatGPT to do that for me. The important role which I use ChatGPT is to review code or troubleshoot errors. So I am not very much familiar with the programming languages like R or Python, but I can use ChatGPT sometimes to generate a code for me. Or if I have a code, I can ask it to troubleshoot if I'm facing any issues. And if I have a manuscript, if I have a draft of a manuscript, I can ask ChatGPT to refine it again for the sake of language or using more scientific words in my literature. And I can also respond to the reviewer's comment, like if I have to write a rebuttal, I can ask ChatGPT what could be the best way to please the reviewers. So that's how I use ChatGPT in my research work. I use AI a lot in literature search, and with that, I like ChatGPT. You can ask a broad question; it will give you relevant information. But I use Perplexity, which will give you an information about the question you ask as well. It can provide you the references, the relevant references, like from which research article or review article that information has been provided. So I use Perplexity to get the relevant literature and the proper citation. And I source this information to another tool called Research Rabbit. And I can, like, if I have a research article, I can get a network of research articles centered around that research article. And I can go through the abstract of these and choose the best article which could be fitting to what research I want to do or what review I want to do. So this is how I use the AI for literature search. Apart from these two programs, I would also like to mention AI research assistants like Elicit and Notebook LM. So if you have not used Elicit, it is a smart paper discovery. You write your biological question. For example, what are the genes which are involved in heat stress tolerance to the plants? I just write this question. So what Elicit will do, it will search for like 50 research articles, and it will choose the best 8 to 10 research articles. Whatever I'm talking is about the free version. If you can pay, you can get better services, of course. So it will choose the best 8 or 10 articles, and based on that 8 to 10 articles, it will give you a summarized review of your biological questions. It can give you tables. It can even identify what were the gaps in the research or what could be the relevant questions which could be followed in the field. This is best for doing a systematic review and meta-analysis. And when I'm talking about the results, apart from getting a complete review of the literature, you will get a table like this, where you will have the study, like from where this relevant literature has been taken from, what kind of study was conducted, what was the organism studied, what were the method used in this study, and the key genes identified. So you get a table, which is quite helpful for a beginner. So this is how I use the Elicit, and once I get the information, I can download all these research articles and feed them to Notebook LM. So what Notebook LM is going to do is, first of all, I have to upload the articles or a document as a query. And if you want a summary of the articles, or if you want to generate hypotheses from these articles, you can use Notebook LM. One of the features which I like most is that it can generate an interactive audiobook for you. So it can generate like a summary of all the articles, which you can download, and you can listen to it even if you are driving your car, or even if you are doing anything and you want to just play it in the background, you can do that. So if you want a summary of the article and you don't want to read the articles, you can generate the audiobook and read them whenever you have the time. This is best for writing articles and grant proposals. And one of the features which I like most is generation of a mind map. So for example, if I ask the question how you can use CRISPR-Cas9 in plant biotechnology, so there will be sections like introduction, CRISPR-Cas9, CRISPR-Cas9 mechanism, CRISPR variants, etc. And you can click it further to get more information. So it's kind of a flow chart, and in that flow chart, you can read the complete article. Oops. So how I use both of these platforms is that I use Elicit to find the papers, I feed them to Notebook LM to analyze them and give me a summary. I use AI for data interpretation. For example, I am a wet lab biologist, and when I have to start working on the bioinformatics part or the dry lab part, I was doing an RNA-seq analysis for which I had to make the PCA plot, or I have to make a volcano plot, or I have to generate heat map, or a box plot with the statistical analysis, for example, ANOVA, and I don't know how to do that. So I can just download this image, one of these image, and feed it to ChatGPT and ask it, please generate a R code to make a similar plot. That's what I did. I took this heat map and uploaded it into the ChatGPT and asked it, please generate a R code to generate a similar heat map. And ChatGPT is going to give me a R code. I would like to mention here that you should be knowing to understand like how to use this R script and how to troubleshoot them. Even for troubleshooting, you can take help of ChatGPT, but as Marina told, that you have to compare the results generated by the AI program, and also you have to use your intellect. So how we can use the AI? We can use the AI for skill building and learning. So AI can provide tutoring platform, and it can simulate a kind of a mentor or a teacher. It can offer you guidance in coding, like learning Python and R. There are like a lot of programs through which you can learn the coding. If you already know the programming language and you have a code and you want to understand if something is going wrong in your code, or if there is a package in R which you are not able to install, you can take the help of AI, which can mentor you to run through that complete process. If you are new to statistics, you don't know which statistical program, statistics you have to apply on your data, you can ask ChatGPT, okay, I have this kind of data. Should I run t-test? Should I run ANOVA? And ChatGPT is going to help you out on that. And even you can ask for R script or a Python script to run that on your data to make a plot along with the statistical analysis. What helped me a lot is that sometimes when you are a wet lab scientist and you want to start dry lab things, you might not understand all of the parameters of the pipeline you are running. So you can walk through the bioinformatics pipeline step by step by using ChatGPT. Like, for example, if you are working on a particular organism and that genome could be different from other organism genome, you can ask ChatGPT what kind of parameters you need to use for the analysis. So in conclusion, biologists should not fear AI, but they should partner with it. They should try to incorporate AI in their research. And I would encourage everybody to learn coding, learn bioinformatics, and use of AI, but in an ethical way. So there is a lot of buzz going around like unethical use of AI, where people are turning research articles or review articles based on AI. So you need to learn how to ethically use the AI. With this, I would like to say that AI should spark your curiosity to learn, but not to silence your critical thinking. So your human intellect is required while you are using the AI. And with this, thanks, and thanks for your patient hearing.
Excellent presentation. Thanks, Dr. Manish. I think we can all take these ideas to improve our processes in the lab, right? Well, I'm the last moderator. I will be running the Q&A session. My name is Jerry Gonzalez. I am a professor of plant physiology here in the University of San Carlos of Guatemala, and I will be running the Q&A session. So I will like to ask the attendees to please type your questions in the Q&A box. We will have a little short Q&A session. So the first question that I would like to ask will be to Dr. Pau. Dr. Pau, do you think it is possible to use image processing to analyze the density of veins in leaves, differentiating their order according to their branching, maybe main, secondary, tertiary veins, etc.? And what programs would you recommend?
For analyzing leaf patterning, well, that's a very good question. Probably there's a software that has, I think that there's a code for that, but I don't have it in mind. But I would say that yes, it should be possible. And yeah, I'm sorry that I cannot, it does not come to my mind, the software indeed, but this can be analyzed. It's a pattern, right? So the venation pattern is a pattern, and you should be able to use AI tools to characterize this. Yeah, absolutely.
Thanks. Here's another question that is that really goes about like the ethics, and maybe Dr. Manish can speak a little bit about how does ChatGPT keep your data safe? Can you upload your manuscript, being sure it will keep it safe?
So I would not suggest to upload a complete manuscript, because AI use that to train the models. So that information could be like available for future users. So until you are very much sure that you are on the stage of like publishing, I would not suggest to upload a manuscript.
Okay, fair enough. I think we should all be like really be precautious about like what type of information we upload to ChatGPT. There's another question for Dr. Marina. Dr. Marina, how do you think AI can help us to do measurements and do studies about abiotic stresses on crops?
Oh, yes, thank you so much. This is very interesting and absolutely relevant question. So it's all about the practical way to take imagery. It can be taken using any type of devices which is available. So it's not just that you need to go and buy some very expensive camera. You can do it with whatever you have. Even the RGB photos taken using a smartphone are relevant for understanding and seeing the stress. But the only very important aspect which I mentioned in my presentation is about the protocol. You need to follow the protocol, specific protocol, like think about light, where are you standing, how far from the plant. So yes, it's very important to collect these images in good quality, and then the model can recognize the early stage reaction for abiotic stress in plant.
Great. Okay. I think here is another question for Dr. Manish, but maybe like anyone can jump in. Do you deal with hallucinations generated by AI tools? How do you figure out plagiarism in summary generated by AI tools?
So as Marina told in her presentation, you have to compare the results across different AI programs. And as I also iterated, you have to use the human intellect always there in the AI, like you have to be careful. And I would not suggest you to use any information, like, I mean, you can use AI program to better your things, but not use AI to generate things. Like if you are talking about the publications in in journals, so you can use AI to better things up to the extent that it's obviously not in the range of plagiarism. So like there has to be a cautious use of AI, and that's where everybody needs to learn about using ethically using the AI in like research writing.
Yeah, I think it's important to set some guidelines about how to use it ethically. Marina, were you going to say something?
Yes, Jerry, thank you so much. I just want to add a bit of practical experience to Manish, what he had said. Absolutely agree, because, for example, we have tried, we did one feasibility study, and it was about apple scab and how can these existing tools, which Manish have demonstrated, how they can help us to understand the amount of, let's say, chemicals which should be applied to prevent the apple scab spreading. And we put, as an input, we put the prompt with very specific data, with numbers, with grams, with everything. But as an output, the result, we got quite generic response. And this is the problem, that you cannot rely, and especially you cannot risk your harvest and your orchard because you will just read that in the generative AI system. So if you are trying to apply something, just double check and apply your brain, the correct thing.
Great. I have one final question that's directed to Dr. Marina, but anyone can jump in if you have something that you would like to share. Dr. Marina, you're based in the UK, as I am, so I was just curious as to whether your work solely with researchers or with farmers as well. Have you had any trouble convincing farmers that your technology can be useful for them on a large scale, or is your work primarily on small scale studies?
Thank you so much for this very interesting question, and I can say that we can spend 1 more hour just to discuss it because it's honestly a very hot question. But as of now, what we can see that because of this, let's say, bias existing regarding AI applications, so we have quite a few groups of farmers who are early adopters of technologies, and they are curious about the possibilities. They are interested in improving their productivity, and they have the farm and they have read that AI can help, and they want to mix it to see the result. So you're absolutely right. In most of the cases, it's all about the small scale application because it's like a trial application. But I'm sure that after a few years, because as I mentioned, AI has just, it's on the rise. So I'm sure in a few years, we will see the more wide scale application in practical level.
Thank you very much. So I think we can close up the Q&A session, and I'll hand it to Indrani.
Yeah, thank you, Jerry, and thank you so much, Dr. Jordan, Dr. Kuzmanko, and Dr. Manish, for your insightful speeches and talks. And we really got to learn a lot from this webinar today. And we have actually had feedback from our viewers that they would have wanted this to last for another 30 to 45 minutes. But unfortunately, that's how this webinar is set up. Like, we have only 1 hour, and we had a lot many questions. We have our socials, our speakers' socials given on the Q&A chat box. I think one of the Plantae fellows can again go and list that out on the chat box for our viewers. So if you have any more questions, please get in touch with our speakers over email or over their socials on LinkedIn and Twitter, and just search them. And I'm pretty much sure they'll be happy to give you their valuable insight on this topic. So thank you all. Thank you, my Plantae Fellows team, moderators team. Thank you, Jason. And with that, I would like to end this webinar. Thank you for your time and grateful for being here.
Thank you everyone for joining, and we hope to see you in the next webinar. The recording will be at the webinar blog, which we put in the chat. So stay tuned and come back. And if you want to rewatch, it'll be there in a couple of days. Thanks everyone, and have a great long weekend for those of us in the US, and great weekend for the rest of us. Bye everyone, and see you next time.
Bye. Thank you, Marina.
Thank you.